I think the dream for us is to make a synthetic kidney either in a chip or in a mini kidney organoid system from stem cells outside the body, where we could use computers and AI to direct light activation.
This is the Discovery Files podcast from the U.S. National Science Foundation. I'm Nate Potker. Kidneys are essential for keeping the body functioning by cleansing the blood, maintaining fluid and electrolyte balance, and helping to regulate blood pressure. But modern diets and lifestyles stress the kidneys, which, unlike the liver, bones, or skin, cannot fully regenerate or heal themselves. The U.S. Centers for Disease Control and Prevention estimate that 1 in 7 Americans suffer from kidney disease, which is progressive and incurable. We're joined by Alex Hughes, Assistant Professor of Bioengineering in the School of Engineering and Applied Science at the University of Pennsylvania, where its lab group is working to understand tissue development and synthetically reconstruct new tissues and regenerate organs in the body. Professor Hughes, thank you for joining me today.
Of course, great to be here.
I'd like to start with a little bit of the background, and I'm curious how you got interested in biological engineering.
Right. So actually, I trained as an undergrad in chemical engineering, and my interests were trending towards biology, but not quite there yet. And I was lucky enough to get admitted to go to grad school at Berkeley in California to do bioengineering PhD. It was a combination of being drawn to, but being good at building microfluidic systems. So a microfluidic chip is something where you can manage fluids at very small volumes. And those types of tools are useful for studying cell behaviors or responses of biological cells to toxins or environmental inputs or signaling inputs. And so I think naturally, after I finished my PhD, moving into a postdoctoral position, I started thinking about, okay, how can I expand my interests to studying cell behavior and especially collective cell behavior? So simple organisms or simple cells are doing amazingly beautiful and complex things together. And yet, our knowledge about how they're communicating, how they're coordinating behavior is quite limited. And so later that developed into an interest in developmental biology, which is really a playground for studying those types of things. And that's led to where I am today.
So I'm going to ask you to elaborate on that term there. What is developmental biology?
Yeah, so it's a broad field, but generally people are interested in studying how organisms come about. So early on, you have to start from a single cell and build all the different cells and all the different structures in the body over time. The question of how you get from that single cell to many and how they structure themselves in the right way to do something functional is just amazingly complicated, beautiful. I think what draws people to it is just, again, that it's a playground for studying these complex construction processes that cells do to take a tubule and branch it and change the cell types in different places and create different functions in the heart than exist in the gut or the liver or the kidney. So there's like the beauty of that process, and there's also connections to other fields like cells often in development. They're very invasive. They move around a lot. So there's connections between developmental biology and, say, cancer. And so a naive way to think about cancer is that it's, in some sense, cells adopting developmental behaviors that they shouldn't have.
And this is kind of where the stem cells people have heard of come in, where
Yes.
It starts off as one thing and maybe becomes something else, and maybe you can make it become something else under the right influence.
It's interesting because stem cells are a big part of explaining how this is possible. You need cells that are naive enough, so they're not they don't have such a strong identity that they can't be turned into heart or turned into kidney or turned into liver. And so a lot of people are interested in taking stem cells. Say you take a skin cell from an adult and you put it into a state where it's suggestible, right? So I can suggest for it to become liver or suggest for it to become kidney. We might see that, okay, for a cell to become a kidney cell, it has to see these 3 signals in this order. And we infer that from the embryo, and then we try to replicate that on stem cells in a culture outside of the body. And we cross our fingers and hope that that's what we see. And that's been remarkably successful. So the stem cell community and the developmental biology community have always been very, very close, if not the same, essentially, same community.
You mentioned kidneys, and your work with kidneys is kind of what brought you to my attention here. Can you talk about broadly what's the kidney's role in the body?
Yeah, so the kidney, it's actually has a range of different functions. So the first function, right, is that it filters blood, and it allows the body to get rid of certain waste molecules. And a lot of its focus actually in doing so, once you form filtrate, is that you want to grab back a lot of useful things, predominantly water. And so the kidney is really this amazing transport organ that has a filter that's followed by very long tubules that are reabsorbing and adjusting the pH and salt balance of the urine. But it also has some kind of other intriguing roles. And by no means I'm not an expert on adult kidney biology because it's very complicated. But it has some really interesting roles in, for example, blood pressure regulation and other hormonal processes. So it has a very strong role in adult physiology and unexpected connections. So injury to the kidney is very common. Chronic kidney disease is very common, and these affect many people.
Millions and millions.
Millions and millions of people. Yep.
So I want to get into your studies a little bit. And one of them is really focused on understanding kidney structure. So can you kind of talk us through that a little bit?
Yeah, I mean, I'm not really sure how I ended up being so interested in kidney. It took a while to decide. And I think what really captured my imagination are the movies that people have made from mouse kidneys as they're developing. Those movies are so beautiful. I wish we could maybe we can show like a movie of one of those, but it really just caught my imagination. And there are so many processes happening all at once in a coordinated way that build the kidney, which is effectively an organ that's just packed with tubules. It's a transport organ, right? It's all about managing fluid. And so as you might imagine, there's just tubules stacked together like in a water treatment facility or a freshwater plant or something like that, right? One thing I didn't know before working on the kidney is that it's quite varied. People have quite a wide range of kidney function just in the kind of just walking around without having any disease per se. One issue with the kidney is that there's this variability, but there's no potential after birth, more or less, for the nephrons, which are these filters, to multiply, right? So you can't build new nephrons as an adult. You're kind of stuck with what you have, and hopefully it's good enough, and hopefully it's your function is high enough that you don't run into issues with disease later on.
So moving into the other study that we're kind of tied together, you're working at growing more of these tissues.
Yes.
How is that possible? What are you working on there?
Yeah. So, as you can imagine, for such a complex organ, there's actually multiple stem cells involved. And we talked briefly about what a stem cell is. It's these cell types that can become different daughter lineages. So it's known that you need, at least 3 stem cell types in order to build something that might look like a kidney. And one of our areas of expertise is in very precisely patterning groups of cells. And we can make many groups at a time in different compositions. So I could take, say, 5 stem cells in one type and put it with 5 stem cells in another type and build a little small community of cells that can interact with each other, signal to each other. And our hypothesis was that by changing that composition of the stem cell types that are there, maybe we could create many kidney tissues that have different properties or different structures. And it's really the beginning of a much longer road in that direction that we can control through engineering. But some of the first things that we saw was that we could take stem cells that give rise to nephrons and put them with stem cells that give rise to the piping of the kidney, the drainage network of the kidney. Just by starting with different compositions, we could create mini kidneys that have quite different composition of the different tubule types in the kidney. So we were able to, in a sense, make little designer tissues and move the needle about the decisions that they're making and what particular structures they make based on that initial condition that we set.
And maybe get a little insight into that role and how it kind of comes together to make them so broadly different. What are the challenges getting those stem cells to work with each other and form the shapes you want them to become?
Oh, it's very challenging. And the students, by no means am I an expert, and the students just have this wonderful capacity to learn and to learn by doing as well, because these cells are very sensitive, as you might imagine. Because they're so suggestible, it's almost like if you look at them in the wrong way, they'll become a cell type that you didn't want, right? And so you have to have very good skills in the lab to be able to grow them and take care of them and prevent them from becoming structures that you don't want. So in order to go into slightly different lineages in the kidney, there's a little bit of black magic about it. We know what some of those factors are, and they have to be very precisely controlled. So, I just have a lot of respect for students and postdocs in my lab that can do this. And maybe one day I'll actually do it myself.
What is the path for this groundwork to become real world applications or part of regenerative medicine? Like, how does this become something that helps people that are on dialysis or something?
Yeah, so I think one of the big impacts that we can have in the future is to address this, lack of therapies that we have for kidney disease. And you touched on one of them, which is dialysis. And the other is simply just replacing the kidney through transplantation. And both of those are not ideal for a lot of reasons, or just have limited capacity. So there's a lot of patients that can never receive a kidney, even if they need one. We have a huge focus on understanding what's going on in development, using it to build tissues, and then to solve remaining engineering challenges that currently prevent us from taking those functional cultures and putting them in the body and rescuing kidney function when we need to. And I would say, primary among those challenges, again, it comes back to plumbing. Like, how do we take the different pieces of the kidney that we can make and put them together in such a way that you have the right connectivity and flows and coordination between what the cell types are doing. And that's where we think this cell patterning approach can help. Another one, interestingly, is the vasculature. So this is the blood flow. Blood flow is super important to kidney function. And you can imagine that if you want to put in a synthetic kidney, one of these, say, engineered biological kidneys into the body, we need to properly connect it with the blood flow of the patient. And it's easier said than done. But I think some of the work that we're doing is pointing in that direction, whether we like it or not. So we're starting to learn about how vasculature is patterned and integrated with these tissues. So still engineering challenges to go, but 100% our goal is to make this have an impact in the clinic and to rescue function in so many patients that for which dialysis or transplantation is not an option, or leads to loss of quality of life.
I'm imagining that the structure leading into the kidney is probably also pretty unique from person to person.
Yeah, actually, there's not much known. So this actually, you would think that human development is, it's like if we've mapped the internet, we must have mapped human development. That's just not the case, right? Because you can imagine that having the right tissues and having the right, it's very hard to look inside the body and see an embryo develop. And that has really hamstrung some of these research into these questions like, okay, is the starting point of the kidney bud, is that just different from person to person? Is the environment having an effect? Like, what maternal microenvironments and aspects are there to kidney development? These things are studied at a clinical level, but very difficult to study at a fundamental level.
Yeah,
it's still a mystery, but the stem cell approaches, those are helping, and also studying in model organisms such as the mouse gives us a lot of information.
Very cool. So I want to ask about other work in the lab. AI is a huge area of interest for people right now. Are you guys using any AI tools in your lab?
Yeah, so it's becoming it's actually becoming hugely useful. So you could use AI tools to basically find structures in, say, kidney slices or cell cultures. And this is very useful because often the task is to understand, okay, what cells are there? what's their morphology, what do they look like, how are they interacting with neighbors. These things are difficult to do manually. And so AI is really helping us to say, okay, this is a nephron progenitor. It's right next to a stromal cell and it has this contact area. So we can do these kind of atlas level studies of cell interactions as the cells are, undergoing these complex behaviors to build tissues. The other area is coding. So it's going to be interesting to see how this plays out, because I'm not saying I'm, at the end of my career or anything, but I'm in this generation that generally knows how to code. And the AI tools can produce very accurate coding to help with tasks that maybe my aptitude is not quite up to yet. And so it's been very helpful for me to, have an idea about a model or a way of analyzing data, having the AI tool help me produce complex code to analyze that. And then I can go and check. At least my coding, my level of knowledge is high enough that I can go and check where there might be errors, right? And so I think for my generation, and hopefully for the next generation of students, we can keep helping people. We can give them new ways to integrate AI into these coding processes that are so important.
So I want to ask you about your career award. How has NSF support impacted your work?
Yeah, so the NSF is really one of the primary institutions in the U.S. that helps researchers with funding to complete these types of research projects. This is very nonlinear, and I think it's really important for people to understand that new discoveries, sometimes they come from unexpected places, right? And so institutions like the NSF are so important for continuing support and funding and creating new technological advances that help many, many people in the U.S. We've received support through an NSF Career Award, and that's really helped to fund a lot of the work that we do in understanding the mechanics of development, how cells interact, what forces do they generate and respond to. I can't speak highly enough about the NSF and what they do to support research in the U.S.
The last question I had for you today is kind of thinking ahead and what's next, or what are the next steps in your work here?
Oh, so many things. And it's just exploding into so many different areas. I mean, we're really interested in, what these geometric differences are between people's kidneys, how to explain that physically. So we recently have become interested in this idea of there being a clock or a rhythm that governs the rate of nephron production in the kidney when it's developing. And so this clock has just been so fascinating to me, because it's not just that cells are doing this coordinated dance to build a new filter unit, it's that they do it at the same time. And there seems to be this kind of governor that says, form one, okay, relax, form another one, relax, form another one. And this rhythm we think is super important for setting that number. So studying mouse kidneys and seeing, can we see proteins and genes that are regulated at different parts of this new rhythm that we're characterizing? There's other projects in the lab that are quite varied. Like, for example, we're really interested in making single cell measurements, like measuring all the proteins in single cells very precisely. That goes beyond the kidney. That's for any biological problem or a diagnostic problem. Other projects that are really directed towards these crucial bottlenecks, like the connectivity problem, like how do we make all of these kidney tubules connect together at the right time in the right place to give you parallel function. That's a big focus in the lab too. One other aspect that we're really excited by is controlling cells. So how can we use light to control either gene expression or protein behavior, so that we can, in a remote way, tell certain cells to do things. That type of remote control could be super useful for kicking cells between different lineages or for inducing certain interactions. So several people in the lab are looking at ways to, for example, optogenetically stimulate branching of the tubule structures that form the kidney. And in another sense, we could also use it to control this clock of differentiation of nephrons. And so that's an emerging area that I'm really excited about.
Like, I think of all of these mechanisms being internal, so the light might play a role is very curious to me.
Yeah, and that's tricky if you're trying to operate inside the body. I think the dream for us is to make a synthetic kidney either in a chip or in a mini kidney organoid system from stem cells outside the body, where we could have that control, and we could use computers and AI to direct light activation.
Special thanks to Alex Hughes. For the Discovery Files, I'm Nate Potker. Please subscribe wherever you get podcasts, and if you like our program, share with a friend and consider leaving a review. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.